All right. Hi, and welcome everyone to the next webinar in our Plantae webinar series. My name is Jason Padilla, and I'm your technical host for today's webinar. Next slide, please. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I'd like to give a special thank you to all our ASPB members who are attending today's webinar. Your ASP membership dues help support making this webinar possible. For any of you who have not yet joined ASPB, you can join today using the discount code PRESENTS10 to receive a 10% discount on your membership sign-up fee. Members get access to a lot of this webinar and a lot of more ASPB benefits and resources. So you can learn more about ASPB and all the opportunities that we provide at aspb.org. Next slide, please. We've had a lot of Plantae webinars before, there's plenty of recordings that are available for you to view. So if you're looking for additional plant science content and really like this one and want to check out more, you can visit our YouTube channel and explore all our exciting talks that we've had over the past couple of years. Next slide, please. Before we get started, I'd like to go over some housekeeping items to make sure that you get the most out of your attendance in today's webinar. If you are experiencing technical issues, please let me know about those using the Q&A box, or you can email me directly at jpadilla@aspb.org. If you have questions for our speakers, which we welcome all of them, please add them in the Zoom Q&A box, and our moderator will be sharing them with our presenters during the Q&A portion of this webinar. If you have trouble connecting or need to leave early, as I mentioned, this is being recorded and you'll be able to watch it later on once the recording is processed. It'll be posted on our YouTube channel, so make sure, again, follow the YouTube channel so you can explore all of the exciting webinars that we've had, and including this one once it's processed. And so without further ado, I'm going to turn it over to our moderator, our Plantae fellows, Fengula and Fateh, who will facilitate today's session. Take it away, Fateh and Fengula.
Thank you, SPB and Jason, for helping us to organize this webinar, and a big thanks to our invited speakers. Welcome to Plantae webinar, Healthy Supervision in Plant Sciences. We choose to talk about a topic that is important to everyone in research, which is how to create a healthy and supportive research environment. As researchers, we often focus on our projects and scientific questions, but it is equally important to think about how the people behind those projects can grow and develop. Today, we'll hope to open this discussion with our speakers. We will start by introducing our speakers and Plantae moderators, and we will follow speakers' talks and QA session. Audience are required to type their question in the QA box, and we will forward them to the speakers as much as possible after each talk. Starting by introducing us, moderators for this webinar will be Fateh Domide Akande, Research Assistant in Maize Genomics team at the International Institute of Tropical Agriculture, and I, Fengula Avgeri, a postdoctoral researcher at the Department of Medical Biochemistry and Biophysics in Umeå University. We have three speakers today with us from academia: Sienva, Juana Kromdijk, and Charlie Anderson. Starting by introducing our first speaker, Dr. Sienva is a research assistant professor and a member of the scientific leadership team at the Institute of Genomics for Crop Abiotic Stress Tolerance at Texas Tech University. Dr. Ha is also a president and co-founder of Vimex Genomics. Sienva Ha earned his PhD in plant biotechnology and has more than 17 years of research expertise in plant molecular biology, biotechnology, physiology, chemical application, plant signaling, and crop development. He has worked in internationally recognized research institutes in Japan and the US. Dr. Ha has a strong interest in developing stress-resilient crops using innovative techniques. During his professional career, Dr. Ha has published more than 50 papers in relevant research fields. In addition, he is an inventor of several patents filed through RIKEN in Japan and Texas Tech University in the US. And our second speaker will be Dr. Kromdijk, who obtained his bachelor and master degree in plant sciences at Wageningen University. In 2007, he was awarded the Alexander James Keith PhD studentship to work with Professor Howard Griffiths at the University of Cambridge, where he studied the effects of light limitation on the efficiency of the carbon concentrating mechanism in C4 photosynthesis. Following postdoctoral stints at WUR Greenhouse Horticulture and University of Illinois, Wannes returned to Cambridge in 2018 as a research group leader, and currently he is leading the Environmental Plant Physiology Group at the Department of Plant Sciences, and he is a fellow at Pembroke College. Wannes is a UKRI Future Leaders Fellow and the recipient of the SEB President's Medal in 2020. His research group employs a range of techniques from ecophysiology, mathematical modeling, biotechnology, genetic engineering to study plant physiology at the nexus of basic understanding of plant function and applications to improve sustainability and food security. And our last speaker will be Charles Sanderson, who is a professor and associate head of research and faculty success in biology at Penn State University. He completed undergraduate studies at the University of North Carolina at Chapel Hill. After completing a PhD in cell and molecular biology at Stanford University, he became interested in using cell biology to support environmental sustainability. As a postdoctoral researcher at the University of California, Berkeley, he analyzed cell wall expansion in plants. His research group at Penn State investigates the dynamics of plant cell walls to inform the sustainable production of energy, food, and biomaterials from plants. They explore how cell wall components and their interactions underpin the properties and behaviors of plant cells and tissues. Dr. Anderson also chairs the plant biology graduate program, and he is the theme lead of climate and natural systems in the Institute of Energy and Environment. And Fateh, you can welcome our first speaker.
Thank you, Faye, for the introduction. It is my pleasure to invite Prof. Chen Van Ha to deliver the first presentation. We are delighted to have you with us today, and we look forward to learning from your expertise and insight. Prof. R, the floor is yours.
Okay, so can I share my screen?
Yes.
Yes, please, you can go ahead.
Can you see my screen?
Yes,
Okay, can you hear me well?
loud and clear.
Okay, thanks for that. So, hi everyone. So, my name is Jien Van Ha. As introduction, so I currently is a research assistant professor at Texas Tech University, and I also the president and founder for BMX Genomics, the company we formed last year. So, first, I have briefly introduced who I am. So, I graduated from Vietnam National University in Hanoi, Vietnam. And I also graduated PhD in collaboration between VAST and Riken, Japan. So after graduation, so I joined the National Key Lab in Hanoi, Vietnam to work with Dr. Dong Nguyen for a few years. And then I joined Riken and I studied there for 6 years before I joined the Plant Science Center in St. Louis, Missouri for 3 years. And from 2020, I joined Texas Tech University, work at IGCAS, focusing on the plant abiotic stress tolerance. And from 2025, we formed one company focusing on the gene editing technology to improve growth tolerance to abiotic stress and biotic stresses. So here is my research interest and achievement. If you see here, I work with multiple plant species from the mother plants, legume, some cereals, and the fibers. And my stress mainly focus on the seeds and root and the physiologies. Um, my interest is focused on drought, heat, and elevated carbon dioxide. And I also have some project related to the phenotyping. I have genotype to phenotype project. And for the last 5 years, I also developed several new gene editing technology that one of the technology we form the companies. So currently now I got about million in grant and some publication and book chapter and conference. And I also have 5 patents is until now, and then we also have several coming. So in our group, so we have the several directions. So we have the gene discovery regulatory network, with focusing on the gene editing, and also we also do the transgenic approach. We work with multiple crops like sorghum, cotton, soybean, and maize, and rice. And others, we focusing on the signaling molecules and microbiome interactions that we also developed some project currently. And as we are working in Ajika, so I mainly focusing on the abiotic stress tolerance with drought, heat, high salinity, and combination stresses. So talk sharing about how we build and maintain a healthy lab cultures. So from my own experience, so I work in Dickens for 6 years, and in the US also almost 9 years. So as the member of the lab, so we usually I created the lab regulations for all the lab I work in Dickens and also now in Tecatics. So I think that we need to provide a professional and safe environmental working place for all members. This is one of the key I think is success for the labs. And usually this is very helpful for the newcomers when they join the new lab. If we already have the good setup, so they can work and adapt easily. So for the psychology safety, we normalize the mistake as a learning opportunities, especially to the newcomer, and to encourage the high risk, high reward thinking. That is one of the things I would like to share to everyone. So from my own research experience, so high risk, high rewards is thinking is very important. Sometimes, you may think something very different out of the box, and people can think, oh, you are crazy. But it's you have to believe on yourself. So this is one of the things I always sharing to my students. So and based on their own interest, so I always support them for their high risk thinking. So in the lab, we also create a shared lab manual defining work life, boundary, collaboration, expectation, and communication. So in the lab, the communication is very important. So we always encourage the people work in a teams. Of course, we also not limited to the personality, but we expect all the people in the same group. We work together and we publish together. And the inclusion and community, so I established the regular non-academic lab check-ins and celebrate both small wins. For example, whenever we have the grant or publication, we usually have the celebration. So for fostering independence in earlier career researchers, so we set these scaffold autonomy to move from direct supervision to co-pilot to advisory support. It's depending on the time people join in the lab, so they will get the direct supervision from me, and then after a few years they will do a more independent. They can, and then they become advisory for the junior. For the ownership of project, this is one of the thing we always do in both weekend and now text. So in our groups, we allow postdoc and student to define their question, write a manuscript, run proposal, and in some case, we also allow them to be the co-PI or even the PI in their own projects. And The third one is career coaching. So we align daily project tasks along with the long-term career goals. So depending on the direction where they want to go, for example, some pupils they would like to go for a postdoc at academia. We guide them in different things, and we also have some people they prefer to work in the industry after they graduate. So we also do the career coaching for them. And we have one guy they just want to go with the policy, so he took it to the government after he graduated, so we also advise him in different way. So for balancing research productivities and with effective mentoring. So usually we handle the one-on-one meetings for everyone. So of course it will take a lot of time for the supervisors. Like when currently now I have 5 students, and it took me at least 5 hours per week with one-on-one meeting. But I think it's very helpful for the student because they can ask any question, and we can solve the problem immediately if they have. And we delegate and empower, so we involve the senior trainees in peer review, grant writing, and undergrad mentoring. So that I think I already mentioned. So we encourage on postdoc and student to write the manuscript, and especially for the grant. And at Techatex, we have several small grant is directly support to postdoc or students, and that we encourage them to write. And the system over heroes, so this is standardize the lab protocols, data pipeline, and onboarding to free up time for high level thinking. So that's we already do, and then we also doing for the last 9 years. So for the key leadership lesson in growing the research group, so I share for both academia and industry. So I think the most important is high for the fees in curiosity. It is one of the most important. So technical skill, we can toss them, but attitude and collaborative mindset drive the lab cohesion. That is very important. So building a team for the company is one of the most critical steps for growth and success. So personally, we lucky and have very good team built in the company, and we work very smoothly and in the lab. So sometimes we not fail, but we may not we may not have the correct hiring. So some of the project, if we hire the not fit people, so we have to replace it. And the second is adaptable leadership style. So tailor your guidance to each individual working style rather than using one size fit own approach, especially in the company. When I run the companies, this is very important. And then I think when we do the career coaching, especially for the guy who took it to apply for the industrial jobs, we always share this experience. So lead by example. So model healthy and productive work-life balance and academic integrity and resilience to the failed experiment. So as you know, no one want the failed experiment, especially for the PI, because it's waste time, money. But for the learner, like for example, student or postdoc, this is very important. So we learn from the failed experiment and we improve ourselves. And of course, it will take more time and money, but it's very useful for growing in the groups. And then I think this is all my sharing experience. So if you have any question, so I am happy to answer.
Thank you very much, Chen, for that insightful presentation. We appreciate you sharing your perspective with us today. You have highlighted some several important points that I believe our audience will find valuable, especially the part of normalizing mistake for newcomers as learning and ownership of projects by your students. I think that's quite insightful. Another thing is tailoring guidance of based on individual approach. I think that's a key insight there. We have few questions from our audience here, and I would like to go over them. One of them is, as the funding situation can be challenging in plant science, how can new PI who spend many hours writing research grants also focus on supervising students? How stressful is that?
Thank you very for the question. So that is very good question because especially during these periods. So writing grant is always, a lot of stresses, and especially right now, the funding for the research is significantly reduced. So as the PI in the lab, so usually we target with both sides from the industrial grant funding and federal grant funding support. So for federal grant currently now is quite difficult. Even I myself we try like last years I submitted 9 proposals and there's only one succeeds. This is very tough, but depending on your research direction, so sometimes you can find easier and better resource. For example, I usually apply for some community board like sorghum checkup, soybean board, or cotton board, maize board, and peanut board. So we also got several projects from the board, and it's much more easier. And we also got some uh, direct funding from the industry. Like when we do something like we do this POC, the proof of concept project for them. So they also support us to some money to do, and then uh, depending on the how big of the grant we prepare. So some grant I encourage the postdoc and student they write, and I just edit it, so they have the opportunity to learn how to write the grant proposal, and of course to improve the quality. So the supervisor have to involve in that. So and then another like the stress when we grant, but we still have to teach. We also have to supervise the students. It takes a lot of time. So especially in my case now, I have 2 jobs independently. So I have to manage my time, and this is not easy, but I enjoy the uh, working like process and sharing my own experience to the student, and yeah.
Thank you very
But
much, Chen.
yeah, it's stress.
Thank, thank you very much. And then another one is, for a newcomer to your team, how do you communicate your expectation regarding productivity, communication, and professional development?
Yeah, so this is very case by case. So for example, we have the students from very uh, um, from the developing countries where they do not have the opportunity to learn very the high technology. So they completely pure, and it's very easy to train them, but many background we have to spend more time on teaching and training. And we also have the postdoc from very high level of institution, and they have very good experience. So for them, they already have the experience, so we have to treat them differently. And of course, after a few years, we try to have the at least the standard of the lab. For example, after they graduated, at least they have a chance to know everything in the lab, uh, basically, so they can have their own ability to apply for the job in the futures.
hmm
Yeah.
Thank you very much, Chen. Thank you very much. We still have a lot of questions, but because of time, we have to move to the next speaker. As the time goes by, you can probably attend to them in the Q&A section. Faye,
Yeah.
you can take over
Okay.
now. Thank you very much, Chen.
Thank
You can
you.
stop sharing. Faye, you can take over now.
Yeah, and now it's time for our second speaker, Juana Korumdijk. You can share and start, and you're very welcome. to the webinar.
Okay, sorry for the slight delay there. I hope you can hear me okay. You can see the
Yeah,
slides okay?
clear. Yes,
Perfect.
nice and
Well,
clear.
thanks very much for to the organizers for inviting me to this very interesting webinar. It's a topic that I'm not as familiar with to speak about and perhaps some of my research, but I'll do a short introduction and then I'll try to address some of the talking points that that you provided in anticipation of this this webinar. So let me see if I can forward slides. Yeah, there we go. So I run a research group at the University of Cambridge, at the Department of Plant Sciences. We generally focus on environmental plant physiology, but most specifically on photosynthesis. And photosynthesis can be studied at various different scales. And so we try to cover a wide range of different integration levels, as you can see here. So some of my team will study predominantly molecular or cellular processes, whereas others will focus on leaf level phenomena or even all the way to nursery plots and field experiments. I've listed some of our more recent output at the bottom there, in case some of our audience might be interested in seeing that. But the point of this slide is not to highlight our output so much, but more to show that there's, of course, a need for a lot of variety in different disciplines in a team to accommodate all these different skills. So when I finished my PhD, initially I moved into a position at a research institute in the Netherlands, where I worked for 3 years. And then I had an opportunity to join the team of Professor Steve Long in Illinois to work there for 5 years. And when I joined Cambridge again to become a group leader myself, I anticipated that my team would roughly be about 5 to 6 people, maybe 7 or 8 at best. But it grew a lot quicker. So I decided for this webinar to provide a bit of a quantitative overview, just to give an impression for, sometimes you get more than you gamble for. And this created some very specific challenges for supervision, but also just the general dynamic of the group. And so these are the main talking points that I decided to focus on for this very short presentation. Initially, just the supervision practices that support the well-being and scientific success. And in particular, what we focus on in my group are to make sure there's protected time for supervision. So any group member doesn't struggle to get supervision time. Within the supervisions, we focus on conversation rather than productivity assessment. And we really try to create a safe space to mainly discuss challenges during research, which can be research related, but they can also be something altogether different that might be related to, life-changing events that might happen in someone's life, which then affects their ability to do research. Could be anything really. And then, of course, with every specific group member at every stage of their development, we try to tailor the planning of their career and the planning of their development according to where they want to go next. Now, what I wanted to focus on with this talk specifically is how my supervision strategies evolved as my research group developed, grew, and diversified. And I think these are some of the talking points that I'll try to focus on in the next few slides. So, of course, with a bigger group, you have a much increased importance of effective lab management. I was very lucky that while my group was expanding, I also managed to recruit an incredibly competent and efficient lab manager. And I think that's really key also to healthy supervision, because if you don't have that and your lab is constantly in shambles because you don't have effective lab management in place, that in itself is a source for issues. And so I think I just got very lucky there, but that's definitely a prerequisite for healthy supervision, I think. And then as my lab grew and we got more graduate students and more postdocs joining the group, I also realized that it wasn't a good practice to just focus on direct one-on-one supervision meetings. And so instead, we started to work with tiered mentoring, and I'll explain a little bit more about what I mean by that in a minute. We're currently also trying to work more towards working even in sub-teams where we group people by overlapping research interest in their project. So of course, they're all working on independent projects by and large, but there is sufficient overlap to try and put people together based on their interests. So for instance, we work a lot on chilling tolerance and maize. We have several people working on that, and of course they together know more than each of them individually, and that also goes for me. So we try to combine that knowledge and make the best of it. And then at a broader context, that also applies to the whole group. So we try to build group cohesion and also develop more peer support as a result of that. So protected time in my group is organized both through regular frequency scheduled meetings. They're not quite as high frequency as some other groups do this. So we have them every 4 weeks by default. But this is not the only type of meeting. So this is the type of meeting that is providing the scaffold. Typically, when people get started in my group, they have a lot more scheduled meetings, and then gradually you work towards a 4-weekly interval. And this is a two-way street. So this is both for them to see me at least at a minimal frequency, but also for me to keep on top of what goes on with each person in my lab. But in addition to that, I have an open door policy, which I make very clear at the start of people in my group, which allows them to come in with anything that they want to talk about. As long as I'm not in a meeting, I'm very happy to entertain ad hoc meetings. And in fact, I do the same with them. So I also use the office space in my lab for just quick drop-ins and just quick chats with people to keep things relatively informal. And I think it's important to, at least in my philosophy, it's quite important to keep an informal structure. We don't have fixed agendas. We try to have a conversation about current challenges or obstacles in research, typically focused on what went on the last couple of weeks and what might be the planning for the next couple of weeks. But the topics of the meetings can vary dramatically based on what's on someone's mind. Then the tiered mentoring that I mentioned earlier is what I brought in when my group very rapidly expanded from a couple of graduate students and a single postdoc to several graduate students and several postdocs. And I realized that I wasn't making the best use of my group shared experience by doing all the supervisions and all the meetings one-to-one only. So instead, I try to pair up every graduate student with a more experienced member of the team, which then creates a supervisory team effectively. So postdocs often take part in supervision in my group, and this means that we have a bigger base of experience to work with for every graduate student, and also that the graduate students have access to a constant source of daily supervision of someone who's more experienced than they are and is still quite well aware of their project, more so than perhaps other members of the lab. And of course, for postdocs, this also gives an opportunity to build some experience with mentoring. It's something that they can put on their resume. And often they have such intellectual input that they end up being co-authors on papers as well. So it's definitely a win-win situation if done correctly. It's not mandatory. If someone really objects, they don't have to participate, but it's something that is often appreciated. And then peer support is something that we try to install at the moment also by grouping by project focus and also having lab meeting progress update in a very informal way so that the whole group can really partake in giving feedback. As you might have seen from the type of experiments we do, we need to have the whole group involved, and I'll get to that a little bit later, but it's some of the experiments we do, we simply can't do by a single pair of hands. So we need to have a very cohesive group. Group meetings are really important for that. They're, I think, mostly chaired by and organized by postdocs in most groups. That's also the case in my group. And we encourage informal formatting and really cater for responses from the rest of the group. So we don't want a very polished talk that is basically like a conference presentation, but we want something that really addresses a particular challenge or something that's encountered, not so much focused on how much progress have I made in the last period since you've seen me last. Of course, research progress is always a key part of these updates, but we also focus on experimental planning. Manuscript drafting is something that we're doing collectively as a feedback sort of peer review activity. We run journal clubs like I think a lot of labs do as well. And we also do group peer review of manuscripts that are coming in for review. And this is also a bit of training of being good citizens in people, especially wanting to continue in a scientific career. It's a useful skill to have. Of course, practice talks, technique seminars. The key of these group meetings is also to try to not keep them too fixed in their format and mix them up every so often so it doesn't get too much of a chore to attend them. And more recently, we've joined up with another lab as well to have shared group meetings with them to have even a bigger variety of audience and hopefully make more use of the base of the skill set. And then I think finally, as I already mentioned, our work typically involves, well, a lot of our work involves quite large experiments that need a lot of hands. And so we really encourage working together. Science, as it's structured in most places, is quite an individual undertaking. And I think this shared endeavor and the shared doing experiments together can also have real benefits in terms of healthy supervision. So reciprocal help, of course, if you're generous with your time for someone else, you can expect the same in return. Often this also leads to inclusive co-authorship. And also it builds a sense of a group identity and people sort of having fun together rather than on their own and celebrating individual successes rather than joint success or the other way around. So in our case, we also have a variety of skill sets in the lab to accommodate all the different things we do. And I think working together makes much better use of those skills than just focusing on each project individually. And as I said before, the working together enhances the group cohesion to the point that it does effectively function as peer support. So if things don't work so well, if you work together and you're more aware of what the others are doing, you can support each other with some disappointments. And of course, if you have successes, it's also much easier to celebrate them together if something good happens. So if you have a nice publication or some grant gets funded, you can celebrate together. And ultimately, this is also good for productivity. And a key example with that as well is in terms of publications that a lot of us need in order to further our careers. By the time our short-term appointments or positions at our institutions in terms of graduate students or postdocs have run their course, you might just be able to submit a few manuscripts. But by the time the revisions come back, often the first author is already left for a different position. If you then have a team of folks in the lab that have jointly developed this manuscript with you, it's much easier to get some revisions in place by someone else doing a few additional experiments for you while you're already working on your next position. So I think it has a lot of benefits, but it needs to be done in a non-mandatory way, but in a way that everyone's happy with. So I think that's all. Thought I'd stick a picture in there of the folks that actually did the work in my lab and some of the names that don't actually match the picture. So we need a new group picture, but I think I'll stop chatting there.
Thank you very much. This was, all of that were really interesting. And especially about the lab meetings and that we have to be a little more casual about chatting with each other. That also reduces the stress. And I have only one question because we are a little bit wait for time. Since you created sub-teams, how you deal with the final decisions? About papers, projects, publication, experiments, all comes through your hands, or you also share the final decisions with postdocs or lab managers?
So there's always someone who typically takes the lead with writing the manuscript. And so in that sense, it's relatively self-organizing. I tend to be reasonably involved at the later stages and sometimes also at the earlier stages. It depends a bit on how easy someone finds it to write and structure a manuscript. In terms of authorship lists, as I mentioned also in the slide, we try to be very inclusive there. So I think most of the arguments and squibbles about scientific publications tend to come from lack of generosity. And if someone's done something to further a manuscript, then they should be on the author list. And this is also encouraged now institution-wide in Cambridge to make sure that technicians, which are often overlooked in terms of manuscript authorship, also find a place on there. So I think our author lists are perhaps a little bit longer than they used to be, but I think that's generally a very good thing. I'm not sure if that fully answered your question, but I'm happy to engage it further if needed.
No, I'm pretty covered, and very interesting insights once again. And Fateh, you can take the lead now.
Thank you, Faye, and thank you to Juana for the presentation. And now we move to our third and final speaker, Charles Anderson. On behalf of the organizing team, I would like to warmly welcome you and thank you for taking the time to be with us today. We are excited to hear your perspectives. Over to you, Anderson.
Thank you very much, Fatai, and thanks, Fingola, for also co-hosting this wonderful event.
Thank you.
So I'm going to go ahead and share my screen. Is everybody able to hear me and see the slides okay?
Loud and clear. We can see your slide.
Wonderful. So some of the things I'm going to say are going to echo my colleagues Jen and Johannes. So it's great to see that we're thinking along similar lines in some cases. This is a picture of my research group at Penn State. We're in the biology department, and I serve a role as a professor and PI of the group, and also am an associate head of research and faculty success in my department, which means that I help mentor faculty and give them advice about how to run their groups as they see fit. So a little bit about me. I actually grew up here in Pennsylvania. I went to high school in Lancaster, Pennsylvania, and got really interested in microscopy in high school. I was looking at Paramecia enuglina under the microscope and just became fascinated with what was happening there at the microscopic level. I did my undergraduate research actually in animals, working with Drosophila development and cell-cell adhesion at the University of North Carolina, Chapel Hill. I then went out to California and did my PhD at Stanford, studying centrosomes and primary cilia in animal cells again. And then at the end of my PhD, I got very interested in bioenergy and addressing the challenge of climate change, which led me to join UC Berkeley for my postdoctoral research and dive into the exciting world of plant cell biology. I worked with Dr. Chris Somerville there to study cell wall expansion, mostly in Arabidopsis thaliana, and I've continued working on the dynamics of plant cell walls for sustainability in my lab here at Penn State, which started about 14 and a half years ago. So throughout that time, I have learned a lot about how different mentors and advisors run groups and tried to sort of glean as much information as I could, and I'm happy to share a little bit of that with you today. So a few guiding principles that I use in the leadership of my own group. I try to remember that science is an exciting journey of discovery. It's not always a destination. It's sometimes the journey is just as exciting as the destination. And it's undertaken by teams of amazing people, right? So science, even though we sometimes think of it as a somewhat solitary pursuit or an individual scientist working at the bench, in almost every case, a major discovery is supported by a team of researchers working together toward a goal. The other thing I try to remember with people in my group, regardless of what stage they're at in their training, is that everyone is a scientist, meaning that they can provide new insights and make valuable contributions to our science, even if their knowledge base is not as extensive as somebody who's been in a certain field for a while. And making that transition from an animal biologist to a plant biologist in between my PhD and postdoc helped me realize that. I had to learn basically everything about plants from scratch, starting after my PhD. So I try to maintain that knowledge of learning new fields and how exciting that can be. I also try to distinguish between supervision and mentorship. So for me, supervision is, can you still hear me okay?
Yes, clear.
Okay, great. So supervision and mentorship are somewhat distinct activities. Supervision is often what we do with the people in our group, but mentorship we can provide to people outside of our groups, maybe to students whose committees we serve on, or to colleagues in different labs that are postdocs and sort of moving toward leading their own group. And that mentorship can be really fulfilling both for the mentee and the mentor. And then leading a research group, in my view, often involves having some kind of vision or direction. That's where my sort of Moana-inspired diagram on the right is, right? That there's somebody who's helping to navigate and guide that group of people through the ocean towards some destination. As a PI, that's part of what I see as my role, is to help envisioning the research directions that our lab is taking and guide the team to make that vision a reality. That doesn't mean that we dictate everything that happens. And some of the most exciting discoveries that have happened in my lab have happened from students and postdocs rather than from myself. And then the other thing I would emphasize, as my colleagues also emphasized, is that having a good, healthy work-life balance is really important, I think, for doing great science, and that PIs can provide a good example of this balance. So that's something I try to remember whenever I think, oh, I have, this grant proposal to write, but I also have family obligations, and I try to strike a good balance between them. One of the things that I also encourage people to do when they're getting ready to start their own lab, I'm lucky enough to have a postdoc in my lab right now who's leaving next week to start his own research group at a university. So we've had some great discussions about this, is establishing what your lab culture will be. And that doesn't have to be the same as what your PI from your postdoc lab or PhD lab had as their lab culture. One of the things that's really helpful for doing that is to develop a mission statement for your group. In many cases, when you're starting a lab, that might be a mission statement that you draft yourself. But what I typically do now with my lab is we have a sort of state of the lab meeting every year, and we go over our mission statement and think about it and think about whether we should revise it or add or take away some of the language in there so that it matches what our mission is as a lab, our scientific mission, and how we go about doing our science. And that leads into the second point, is that talking about how we do science in the lab with prospective group members or current group members, that can make a big difference in sort of maintaining and enhancing that lab culture. And then demonstrating and praising positive behaviors and practices in the lab, and correcting unproductive actions with clear explanations and pointing out that these are learning opportunities rather than things to be afraid of, I think is really important, right? So again, Chen especially pointed out that taking risks is part of science as long as we do it in a safe way, right? In a safe context, that can be really productive. And then just there's a copy of my mission statement. I'm not going to ask everybody to read it, but if you're watching this as a recording, you can pause it. How to help trainees master research skills. Many research skills are fairly straightforward. Pipetting takes a few minutes to learn, but to really get great at microscopy, for instance, does take weeks or months or sometimes years of training and practice. So one thing I like to emphasize for people in my group is that developing leading expertise in a specific subdiscipline, in my case, it's mostly cell biology and imaging, can be very effective scientifically. You can enhance your group's reputation in that particular area. Collaborators will seek you out. People might ask you to write review articles about that particular topic. So as a cell biologist, we often try to be jack of all trades, mixing biochemistry, molecular biology, genetics, structural biology, physiology, all of those things together. And we try to apply those to the scientific questions that we're asking, but really having leading expertise in a particular area can help a lot, right? That really opens doors, especially in terms of collaboration. Taking the time to work directly with your trainees at the microscope or the bench or the computer, depending on what discipline you're working in, that can really help people learn from mistakes that they might make. It might help you discover something new by discussing what they're seeing on the microscope or on the computer or at the bench. And that will really pay off overall in terms of empowering the people in your lab to become independent. And then encouraging iterative mastery of complex skills like experimental design and data analysis or interpretation, I think is especially useful. Not saying you have to master this all all at once right away, but that you're building this skill over time to become an expert in this particular area. And then being honest with yourself about what your strengths and weaknesses might be as a scientist, teacher, or mentor. I am a really good cell biologist and microscopist in my own view, I enjoy writing and I'm a strong writer, but I'm less strong in statistics and bioinformatics. So I seek help from colleagues. I point my trainees in the direction of colleagues here at Penn State who have expertise in those other tasks. Another thing I would echo from Chen's presentation is persevering, right? And fostering creativity. I think Johannes also said some really nice things about how to foster creativity in group meetings by changing the format and having sort of open discussions. Creativity is one of the most important things we bring to science, and it's something that we sort of have to foster and nurture and build as mentors. So talking about the details of experiments and results, often I meet with everybody in my lab once a week. We also tried an experiment with subgroup meetings, similar to what Johannes mentioned this summer, and that worked out really nicely. But also, sort of stepping back and surveying the big picture with your trainee, that can provide useful perspective. And that's something that trainees sometimes miss because they are really focused on the details. The details matter a lot, but also taking time to step back and look at the big picture, I think, can be really helpful. Demonstrating patience, right? That's one thing that I don't naturally have a lot of. I'm a pretty impatient person when it comes to science. I'm like, let's get this result right away. But having patience to maybe, do that set of plant crosses or set up this tool that might give you a result in the future can be really critical for achieving a larger goal than just, going for the quick and easy result. Facilitating discussions within your group, between groups, and with the larger scientific community, those are often a source of new ideas. And that's something that I really encourage people in my group to do, to reach out to, open the door to potential new collaborations. And then encouraging occasionally, at least, some blue sky thinking, right? Let's think about what we might do as a lab. So one of the things that I have as a goal this year is to set up a lab retreat so we can talk about some of those like longer picture ideas and how that might fit into existing knowledge, feasibility, and resources, how we might advance our science effectively. One observation that I've made about trainees is that there are some scientists, some trainees that tend to be really deep thinkers. They read the literature extensively, they think a lot about their experiments, and that's super useful. Others are more action-oriented, right? So instead of like the thinker sitting and thinking, they're like the Hulk. They're like, let's smash this science and get it done. Both of those things can contribute to good science, and I think some of the best scientists I've met have a good balance of those aspects, right? They might be thinking while they're, cranking through some tedious task in the lab, and that can really help them enhance what they're doing. So I encourage some level of mindfulness in the science that's going on in our lab, and that means thinking while you're maybe collecting microscopy data or analyzing those data. That doesn't mean you need to only be thinking and talking about science, but most of what we try to do in the lab is use our time for science time. And then we also have time for social interactions. Most of my group gets together for lunch every day and they chat about all sorts of things, and that's great. As a leader, I actually intentionally don't join those meetings, not because I don't like the people in my lab, I like all of them, but because I want to give them the space to speak freely without feeling like I'm looking over their shoulder. We talked a little bit before about guidance versus independence, right? Sometimes new trainees are afraid to make mistakes. We can encourage people to take reasonable scientific risks and model learning from failure, right? Learning from failure is a lot of how we do science and make new discoveries. Guidance in the form of, maybe daily feedback when somebody's starting out can help reduce uncertainty and maybe the fear of making a mistake. And it can foster useful creativity by placing some constraints on the ideas and strategies we might use to address something, right? So we don't want to necessarily have a completely open box, but maybe somewhat of a box that people can work in and be creative in that space. And then encouraging independence while providing support. So we say, I want you to go do this, but if you need help, come talk to me. I also have that open door policy, like Johannes mentioned, that can result in self-efficacy for scientists where they feel empowered to then do the things that are going to get to that next step of knowledge or make this next discovery. So a scientific neophyte and a beginner might need some supervision, whereas an independent scientist needs less supervision and therefore is empowered, although they still benefit from having discussions. And then the image in the upper right by Pavlović is just the actual Greek character mentor, mentoring one of the characters from the Odyssey, which is a hot topic right now culturally. Okay, so I'm going to finish up there. And I'm going to stop sharing and open the floor to questions.
Thank you very much for the excellent presentation and the scene. From the fact that everyone is a scientist to the need to nurture creativity and building and encouraging independence while providing support. It was a wonderful presentation. Thank you for your time. The first question from our Q&A session is, after many years of supervising students and being your role as a faculty success, what qualities do you believe define healthy and effective supervision? That's the first question.
Sure, that is a really good question. I would say that maintaining curiosity as a PI is something that's extremely important, right? You want to be super excited about all the science that's happening in your lab. That's not very hard for most professors because we got into this field for a reason, right? We got into this job for a reason in that we are naturally curious and inquisitive about science. But I think just demonstrating that enthusiasm, sometimes that can be challenging when we are like stressed out from writing too many grant proposals or we have something going on with our families at home or something like that. Those are things that we sometimes have to filter a little bit as leaders of a research group, and that's okay too. Not to say that we should hide what's going on in our lives from our trainees and lab members, but they are here to do science. They're here to learn from us and to benefit from our expertise. And that's really our role that we play when we come to the lab. So that's not to say that we can't bring ourselves, but I try to bring the best version of myself to my lab. And sometimes that's a little challenging because I, again, might have other things going on. So yeah, that's one answer.
Exactly. You're absolutely spot on. For an established faculty member as yours, how do you maintain supportive research environments while managing increasing leadership responsibilities, especially the administrative responsibility when they started coming in? How do you maintain the balance?
Yeah, that's a really good question. I think about that a lot in conversations, for
Because
instance,
you are the
with,
program director, you are faculty
yeah,
success, then you still have classes to take. How do you maintain the balance?
right. It's a great question. And one of the things that I try to do is protect that time that I'm able to dedicate to research. So I have weekly meetings with my students and postdocs every week, and I really do my best to protect that time as long as it's not, a family obligation or something like that. I try to say that's the priority for me on that particular day of the week. I try not to schedule meetings during that time if I can at all help it. I think it's also important to recognize that as a faculty member, you're not just constantly building up the number of things you do. Sometimes you might shift directions. If, for instance, I was recently the co-director of a center here at Penn State, and I stepped down from that role after I became the graduate program chair so that I could balance the time that I was dedicating to service with my own research and teaching obligations. So I do think it's important that as we sort of, progress in our professional careers, do maintain some level of balance between the research, which really is exciting to many of us, the teaching that is also exciting and provides ideas for research. One thing I would point out is that teaching is an amazing place to learn, new ideas that might be applicable to your research if you're teaching on a topic that's not your expertise. And it's also a great way to just have a lot of good conversations with excited young scientists. So I love teaching. I kind of miss teaching, and some of my service roles take me away from teaching right now. But that's another thing I would emphasize is just try to maintain that balance of teaching, research, service, depending on what your university asks of you.
Maintaining balance, I think that's key. One final question, and I think this involves both you and Wana. And the question is, Wana mentioned focus on conversation rather than productivity, while you mentioned correct unproductive action with clear explanations. Could you both explain more about your guidelines and criteria for productivity? Did you get that?
Yeah, I'll jump in first and just say that for me, productivity means advancing knowledge in science. It doesn't necessarily equate to you must have these many papers published in these types of journals by the time you graduate or complete your postdoc. Ultimately, we want to publish everything that we do as scientists because in many cases we're funded by the public and we want the public to benefit from the science that we do. So one of the ways we do that is by publishing our work and sharing it with the community writ large. For me, productivity means that you're making forward progress and that you're addressing challenges as they arise, not necessarily that you're getting a specific result. I actually was a rotation student during my PhD in a lab where they rewarded certain specific results with like a lab party. And that was cool in some ways. It was nice to be like, yes, I finally did this. But it was also a bit risky in my view because it encouraged people to fixate on a specific result rather than just getting a lot of good other data that might address this hypothesis or disprove that hypothesis. So that
Mm.
was always a little bit of a danger zone to me, and I don't do that in my own lab. We just have parties as a matter of course. Go ahead, Johannes.
Yeah, I mean, I think Charles covered a lot of the question already. I think what I meant by productivity, maybe this is somewhat counterintuitive, but a lot of the people that I get into my group already come in with a very strong focus on being productive, and to the point that it's sometimes easier to get productive by just having an open conversation about what goes on and not so much focus on the imminent output that they want to achieve. Um, then even emphasizing that productivity element of having a productive career even more. So I think it's yeah a lot of the people that we get in, and I think this is probably true for all of us, have an internal motivation that drives them enough, and we don't need to encourage that even more by having a very high frequency of progress meetings where we just focus on have you produced enough, this, that, or the other. It's much more beneficial to just talk about the science and what what the current challenges are that they have with their projects. So that's what I meant by that.
Mm.
And I agree totally that rewarding certain results over others is an absolute no-go. But I think that goes without saying.
Thank you very much. As a final note, looking for all of our speakers, as a final note, looking ahead, what changes do you think are needed to strengthen mentorship and supervision in plant sciences? I think, Chen, if you want to go first.
Say it again. I cannot hear. You mute.
Okay, I said looking forward, what changes do you think are needed to strengthen mentorship and supervision in plant sciences?
Uh, yeah, so from my side, I think of course everyone have their own dreams, but they have to stay up with the trend, the science, what we have to figure out. It's similar like nowadays the AI, the involved in everywhere, everything, including the plant science, and many part of the work they can do for us. So now I think the first thing people, I mean, the young scientists need to know what is your interest and how we can use the support from, for example, AI to your own research. For example, now, previous many developing countries, they don't use English as a native, so they very, have difficulties for writing grant or manuscript. But right now, they are very good at writing with the AI support. So I think first they have to understand what they want to. For example, we have several people in the lab, so each they have their own goals. So they have to know what they want. And then second, from what they want, so the supervisors and the mentors they will guide them how to go to achieve their goals. So I think this for the young scientists now is much more easier than long time ago. We have to do many work without support, but
Mm,
nowadays the AI support for data analysis, for even making the project and form everything for you. Of course, it's maybe not true, but at least you have some guidance already, and they have the big
mm,
support already. So now the
mm,
most important thing is what you want,
mm,
and you need to look at the window where you can go and to become independent scientists in the futures. Because if we go for the field that so many people they already dig up, so it's still good, but it's been very difficult to become independent scientists and PI in the futures.
Yeah.
That I think.
Thank you very much, Chen. Juana, if you want to go next.
Maybe I'll jump in just because unfortunately I have to go to another meeting that I'm late for. So one thing I would emphasize is the need to recognize that science is a human endeavor and that it can't necessarily be optimized in the same way that technology is sometimes optimized. So I think what I would encourage us all as scientists to do is to be patient with each other about the speed, the pace of scientific advance and progress that happens. I would encourage us to think about how we can do science in a more sustainable way, both from the human perspective and also from the environmental perspective itself. The science that we do has a fairly large carbon footprint, and we have to think carefully about how we're doing that in relation to maybe the discoveries that we're all making that are also helpful for addressing environmental challenges. And then the last thing I would encourage is for us to communicate the benefits of the science that we are doing with everybody. That's critically important for maintaining societal support for science, and that's something that's eroding in some of our societies, unfortunately, right now, partly because we aren't doing a great job of communicating the beneficial aspects of what we do. And I think that's very important, especially as people who are getting a lot of our funding from the public to share with the public why we do what we do and how it is a good thing. So thanks, everybody.
Thank you very much for joining. Thank you.
I think a lot has been said already, Io, but maybe the final thing to add there is that I would say in science there's a lot of different ways to be successful. And I think historically, at least traditionally, there's a typical type of successful scientist that we all picture and sort of celebrate in awards. And I think if a more diverse type of successfulness could be identified and be more clear that it's not just one way that you can be successful in science, that would help a lot. I think I'll leave it there.
Thank you very much, Juana. So, Jason, take it away.
Yeah. All right. Yeah. Thank you, everyone. I know we went a little bit over, but thank you for joining us today. And again, a very special thank you to our speakers, our moderators, and all the ASP members who are participated in today's webinar. And of course, thank you all for attending. Again, this webinar is being recorded, and we'll post this in our YouTube channel. So if you want to rewatch or once it's uploaded, share it with your colleagues and share all the valuable and important things that our speakers shared. You are encouraged to do that. Other than that, I hope everyone have a great rest of your week, and we hope to see you on the next Plantae webinar. Bye, everyone.
Bye.
Bye bye. Thank you.